This video will cover subtopic 3.4, pedigrees. So in 3.4, here are the understandings that we'll cover, as well as the application that we will cover. So in this video, we'll be going through pedigrees. So a pedigree is a type of flowchart that uses symbols to show the inheritance patterns of traits in a family over many generations. So you can use it to track whether a genetic disease is present or not. So genetics use pedigrees to determine the pattern of inheritance of a particular trait and the risk factors associated with that particular genetic disease. There are particular symbols that are used with pedigrees to indicate certain things. So here we have a blank square, which would represent an unaffected male. Here we have a filled in square that would represent an affected male. So this is a male that shows symptoms of that particular genetic disease. A blank circle represents an unaffected female. Filled in circle represents an affected female. If you see a diamond, this represents the sex of a person who is not known. A line between a square, a male, and a female indicates that marriage or mating has occurred. And in the unlikely event that there are two lines, this would indicate a consanguineous marriage. Now, where there is a horizontal line, you can get a vertical line coming from that horizontal line. And that vertical line will indicate that offspring is a result of that marriage or that mating. So here it kind of shows how those lines all work together. So here we have a male, we have a female. There has been a marriage or mating that has occurred, and it has resulted in children from that marriage. So here we have one brother, a sister, and then there were a set of twins produced from that. So if you have two lines kind of stemming from the same origin, this indicates twins. In this case, there was a female and a male twin. Now, in the event that there are identical twins, they should be the same symbol. So with pedigrees, the way it will typically look is that there will be numbers associated off to the left that will indicate the generation. So, number 1 is the first generation, number 2 is second generation, and number 3 represents the third generation. And then individuals are usually numbered and ordered in terms of like oldest or whatever. Affected individuals here are shaded in. Unaffected individuals are not shaded. So here we have an affected male and an unaffected female. And so here, you can see the children that resulted. So they had a daughter, a son, and another daughter, each of whom got married. This daughter had children with an unaffected male. But had one child that was affected and one child that was not affected. So this is typically what pedigrees will show. So we'll build our own pedigree in the following example. So we're told that cystic fibrosis is an autosomal recessive disease. Talia's brother Jonathan, we're told, has cystic fibrosis, but her other brother Benjamin does not. We're told that Talia's maternal grandparents are both carriers of the gene, and Talia's paternal grandfather does not carry the gene for cystic fibrosis. We're asked to draw the pedigree to represent Talia's family, and we're asked to include each person's genotype as best as we can. So, there's two ways that we can approach our pedigree if we're asked to build one. We can start from the top down, starting with the grandparents and then going to the kids, or we can start with the kids and work our way up to the grandparents. Now, based on how this information is presented in this question, we'll start with Talia and her brothers and build our way up from there. So we can assume that Talia does not have the disease, even though it doesn't expressively say. We'll make the assumption that she does not. Whereas Talia's brother Jonathan does have the disease, so we'll draw a square that is shaded in. And then we're told her other brother Benjamin does not. So we will draw some vertical lines to indicate, that they are the result of their parents having mated. So we'll draw a square to represent the dad, a circle to represent the mom, and then we're given information about her maternal grandparents and her paternal grandparents. So we'll draw circles and squares to represent Talia's maternal grandparents. So there's her maternal grandmother and her paternal grandfather. Now I've left myself a lot of space, so I'm going to draw this a little squished here. And then for her paternal side, there is her paternal grandmother and her paternal grandfather. Now, we have a bit more information in this question that we can use to fill in our pedigree. So we're told that Talia's maternal grandparents are both carriers for the gene. So in this case, they are unaffected. So we do not color in her maternal grandparents. They're not affected. We're told that Talia's paternal grandfather does not carry the cystic fibrosis gene, so he is not affected. So odds are that her Talia's mom is going to be a carrier or could even be affected. So we're asked to come up with the genotype of each person. So since we're told that it is autosomal recessive, we can just use capital and lowercase letters to represent the disease. So we'll, for the sake of distinguishing out the letters, we'll use a capital D to represent the dominant gene for the disease, and a lowercase d to represent the recessive or the CF gene. And we can go in and fill in genotypes as best we can, given the information. So we know Talia's one brother is affected, so we can determine his genotype. He will have two lowercase d's. We are also told Talia's maternal grandparents are both carriers, so we can give them a big D and a little d, big D and a little d. And we're told that Talia's paternal grandfather does not carry the gene. The big D, little d, or sorry, big D, big D. And, we're now asked to kind of figure out what each person's genotype could be, or could possibly be. So assuming that Talia is not affected, she could be big D, little d, or big D, big D. And her other brother is not affected, so we know that he'll at least have one dominant allele. So for the mom, in order for this gene to appear, the mom can either be big D, little d, or she could have had the disease as well. So it's or one of those two possibilities. Now, in order for this disease to show up here, Talia's paternal grandmother, we don't know her genotype, so we'll have to make some assumptions here. Now, in order for this disease to show up in Talia's generation, she would have to be that paternal grandmother would either have to be a carrier or have the disease herself. So in this case, whatever genotype is here for the grandmother, the father is going to be a carrier. And so Talia herself, if we look at the possible combinations here for the mom, this will allow the brother to have cystic fibrosis. And then this means that Talia can either be a carrier or completely unaffected. Same with her other, same with her other brother as well. So here we have each person's genotype, as well as drawn out the information that we have for our pedigree based on what's presented in the question. So in terms of patterns of inheritance, there's two types. There's what's called autosomal inheritance. So, these come from the autosomal chromosomes. And remember that autosomes are the non-sex chromosomes. So autosomal inheritance refers to the inheritance of traits whose genes are found on autosomes, chromosomes 1 to 22. Chromosomes 23 are those sex chromosomes. And autosomal genes are responsible for many inherited genetic disorders. So with those patterns of autosomal inheritance, excuse me, we'll take a little break. All right, I apologize for that. So we can have autosomal dominant patterns of inheritance. This occurs when an individual occurs when the disease-causing allele is dominant and an individual has one or both copies of the allele. So you can think of like Huntington's disease, for example. There is autosomal recessive, which occurs when the disease-causing allele is recessive and the individual has both copies of the allele. So cystic fibrosis. Now, in autosomal dominant pedigrees, there is a pattern of inheritance that is usually present. So usually every generation has an affected individual, and unaffected parents cannot have an affected child. So parents who do not have the disease do not have affected children. Their children remain unaffected. So if we look at the following example here, we can see an affected male, and based on patterns of inheritance, they are having children that are affected. So half of their children are affected, and we expect about 50% of their children will be affected. And then in the next generation, the disease is showing up again. And here we can see that, this child was not affected, so they did not get that gene, and none of their children are affected. Now, in autosomal recessive pedigrees, the pattern of inheritance that shows up there is that you can have two unaffected parents, and remember when we have unaffected parents and it's autosomal recessive, they are going to be carriers, and these carriers can have affected children. So it only occurs if both parents are carriers, i.e. they're heterozygous, and with autosomal recessive diseases, they can skip generations, so they show up every other generation. So we can witness that here in this pedigree. So here we have an affected individual, unaffected male. The two children that they have are both unaffected, but we see here in the third generation that we have affected children now starting to show up. So this disease essentially skipped a generation. And then another hint that it is autosomal recessive. Here we have two affected individuals, and all of their children are affected. Now, have a look at the following pedigrees. Do they show autosomal dominant or recessive patterns of inheritance? So take a second, pause the video, and look to figure that out, and then we'll discuss through. All right, so in the first pedigree, if it's autosomal dominant or recessive, so here we have an affected female having children with an unaffected male, and all of the children are affected. This affected individual has children with an unaffected male, and all of the children are affected, which strongly suggests that this is autosomal dominant. In the second pedigree, here we have an affected male, unaffected female. The children that they have are not affected, but in that third generation, this female who has a child with an unaffected male has a child that is affected. So this son here has the disease. So in this case, the disease has skipped a generation. So this would be considered an autosomal recessive disease. Now, in the third example, we have to look to the second generation because we may not have enough information from that first generation. But here we can see we have an affected female having children with an unaffected male, and they had two sons, one of whom was affected. And in this case, we can't determine whether this individual was affected or not. A line through a person means that they are deceased. So we may not be able to know whether they were affected by the disease or not. This affected male had children with an unaffected female and had an affected child. So here we have the disease showing up in every generation. So it is most likely an autosomal dominant disease. In the last example, here we have an affected female having children with an unaffected male, and they have two children that are affected, and this unaffected female has children with an affected male, and then here, what kind of tips me off as to whether this is recessive or dominant is this female here is unaffected, and here we have an affected male, unaffected female, they have an unaffected son, and the mating between these two individuals results in an affected female. So here we have the disease essentially kind of skipping a generation and showing up in the third generation, which does suggest that the disease is due to autosomal recessive inheritance. Now, with pedigrees, you can determine whether these are done through sex-linked genes as well. Now, with that pattern of inheritance that you can look for with sex-linked genes, with a disease that is due to X-linked dominant genes, sons and daughters are both affected. You have more females that are affected if it is dominant. And affected sons must have an affected mother. Affected daughters must have an affected mom or dad. And 100% of the daughters of affected fathers will have the gene. With X-linked recessive genes, males are more likely to be affected, and it's never passed from father to son. That gene comes from mom on her X chromosome that she contributes, and all daughters of affected fathers are carriers, as an affected father would be the one contributing the affected X chromosome. And with Y-linked genes, all sons of affected fathers will have the trait. So in the following pedigrees, take a second to determine whether they show Y-linked or X-linked dominant or recessive inheritance. So in the first pedigree here, we have an affected female. Get the pen. So we have an affected female, an unaffected male, and then the children that result, we have an affected male and an affected female. The two children here, or these two children here, are not affected, but this daughter here has children with an affected male, and the disease is showing up in all of their daughters. So it would suggest that it is an X-linked dominant um, uh, pattern of inheritance. In the following pedigree, we have an affected male and an unaffected female. And here, they're having son, daughter, daughter, son, and both sons have the disease. Those sons who have their own sons also have the disease, which suggests that it is Y-linked. And in the last pedigree, we have an affected female, unaffected male, and here they have an affected son, but unaffected daughters. The unaffected daughter has a son that is affected, suggesting that she is a carrier and pass that gene on to her son, which suggests that it is X-linked recessive. So with genetic diseases, there are more than 4,000 genetic diseases. Many are caused by Mendelian patterns of inheritance, and the chance of inheriting one allele for any specific disease is small, but to develop the disease, two alleles must be inherited, and the chance of that is extremely small itself. So the introduction of these alleles is through mutations, and so a mutation is a random change to the base sequence of a gene. And there's two main ways to develop mutations: through radiation, so gamma rays and alpha particles, which are radiation from radioactive isotopes, as well as shortwave ultraviolet radiation, that UV radiation, and X-rays. So they can damage DNA and cause changes to the base sequence, change the sequence of those letters. There are some chemicals found in tobacco and gases used in chemical weapons, for example, during World War I, that can also cause changes to the DNA base sequence and introduce mutations. So a random change to an allele that is developed by evolution is unlikely to be beneficial. So all mutations are either neutral, meaning there's no harm done, or they can be harmful. So mutations in genes that control the cell cycle are likely to increase cell division and will result in cancer. Mutations in somatic cells do not get passed on to generations, and somatic cells are basically all cells but gametes. So any cell that is not a gamete is a somatic cell. And so it's mutations in germline cells, basically those gamete cells, that get passed to offspring, starting the origin of a genetic disease. So any changes to the base sequence that happens in a gamete will ensure that it is passed on to the next generation, that it gets inherited. So that is the end of this video for 3.4 pedigrees.